Today we're going to talk about cell homeostasis, and the goal of this video is to explain how cells use the cell membrane to maintain homeostasis through diffusion and osmosis. So first, let's talk about the structure of the cell membrane. The cell membrane is composed of phospholipids, and a phospholipid has a hydrophilic head that attracts water and two hydrophobic tails that repel water. At the bottom here, this is one phospholipid. At the top, you have the hydrophilic head. Hydrophilic means water loving, literally. So this part attracts water. And then the two hydrophobic tails, um, those are made of lipids, which are fats, and these repel water. That's why they're hydrophobic. And you can see that there's two tails. The reason the head is hydrophilic, the reason it attracts water is because the head is polar. And remember, water is polar. And like attracts like. But the reason that the tails repel water is because the tails are non-polar. Similar to the way that you can mix water and oil and they don't mix together. They want to stay separated. That's because the oil is non-polar, but the water is polar. Okay, so this is one phospholipid. When you put a whole lot of these together, you can form a cell membrane. The phospholipids in the cell membrane are arranged in two layers. The heads, the hydrophilic heads, are on the outside, and the tails are on the inside. And you can see that in this image on the top here. So when you put all of these phospholipids together, it will look like this. They can form a sphere. So this would be like a cell. And you can see there are heads on the outside of the cell, and then there's heads on the inside. And in between, you have these tails. So that's what the cell membrane would look like. It's a phospholipid bilayer. It's made of phospholipids, and the reason we call it a bilayer is because there are two layers of phospholipids here. All right, now what's the function of the cell membrane? It regulates transport of materials in and out of the cell. Now, an analogy might help. The cell membrane is kind of like a filter because it allows some things in and keeps other things out. And for that reason, we say that the cell membrane is semi-permeable. Now, what does semi-permeable mean? Sounds like a fancy word. Here's the basic idea. You can ask yourself, can materials pass through this filter? If yes, then the filter is permeable. If no, then it's impermeable. Now, for things that are permeable, for a filter that's permeable, you can ask, do only some materials get through? And if the answer is yes, then it's semi-permeable. So it lets some things through, keeps other things out. That's what it means to be semi-permeable. Okay, so that's the function of the cell membrane. It just regulates what comes in and out of the cell. And it can do that because it's semi-permeable. Now, I told you that you can, the cell wants to try and maintain homeostasis through diffusion and osmosis. So let's talk about diffusion now. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. So in this image at the top here, all of these molecules are very close together in one area. So this would be an area of high concentration. But as they spread out and they move away from each other, and there aren't as many molecules in a single area. In fact, when it spreads out pretty evenly, now that's a very low concentration. So on the left, you have a high concentration. On the right, you have a low concentration. And diffusion is just the process where molecules move from an area of high concentration to an area of low concentration. And the difference in the concentrations in these areas create what we call a concentration gradient. So the gradient part means you're moving from, specifically in the case of diffusion, high concentration to low concentration. Now, diffusion is important because it helps the cell reach what we call equilibrium. Equilibrium is where there are equal concentrations on both sides of the membrane. So here's another image to help you see this. Here's this pink line represents the cell membrane. And you can see that on this side of the cell membrane, there's a very high concentration of molecules. And on this side, there's a very low concentration. So we have a concentration gradient now. We have one end that's high, one end that's low. And so over time, if those molecules can pass through the cell membrane, then they will, and you will reach equilibrium. So if you look on the right now, there's approximately an equal number of molecules on both sides of the membrane. So this would represent equilibrium. Now, there are two types of diffusion. There's simple diffusion and facilitated diffusion. Here's what they have in common. Both are still types of diffusion. So but in both cases, molecules are moving from an area of high concentration to an area of low concentration. Here's the difference, though. With simple diffusion, molecules can pass directly through the cell membrane. And the reason they're able to do that is because they're small enough to pass through the membrane. But for very large molecules to get through the membrane, they need a transport protein. So when the cell uh, has molecules pass through from one side to the other through a transport protein, and those molecules move from an area of high concentration to an area of low concentration, that's what we call facilitated diffusion. So the main difference is whether you have this transport protein or not. The similarity is they both move from high to low concentrations. If there isn't a transport protein, then it's simple diffusion. If there is a transport protein, then it's facilitated diffusion. Now, sometimes molecules are too large to pass through the cell membrane. So if you look in this image, these red dots, they are a little bit too large to pass through these gaps. Okay, so the cell membrane acts like a filter again, and these red dots are too large. So even though right now this side is a high concentration and this side is a low, you're not going to be able to, these red dots aren't going to be able to pass through because they're too large. So what happens in this case? Well, osmosis is going to happen now. So osmosis is the diffusion of specifically water molecules, just water molecules. Osmosis is the diffusion of water molecules across a semipermeable membrane. And here's what happens. The water, which again is a solvent, moves to the area with the highest concentration of solute. So whichever side has more of these red dots. So right now the right side has more red dots. So the water on the left side is going to move toward the highest concentration of red dots, the highest concentration of solute. And so water is actually going to move to the right here until you reach equilibrium. There's about an equal concentration on both sides. So you can see the dots are spaced out similarly on both sides now. And the reason this was possible was the water moved to the other side through a membrane, and that's the process of osmosis. Okay, so now you can actually predict what will happen to a cell when you put it in certain solutions. So let's practice this. Pretend that this yellow line represents the cell membrane. So you have an inside of the cell and you have things outside of the cell. Well, you can actually count what's inside and what's outside. Right now, inside the cell, there are 12 water molecules. That's the blue dots. And outside the cell, you have, I'm sorry, inside the cell, you have 2 of these red dots. They represent salt. Okay, so I have 12 water molecules inside and 2 2 salts on the inside. Okay, let's do the outside now. So outside, you have 8 water molecules, 8 blue dots, and 8 salt molecules, so 8 red dots. Okay, once you've established the amount of water and the amount of salt in this case inside and outside the cell, you can actually predict what the cell is going to do to maintain homeostasis here. Notice there's less solute, there's less red dots on the inside of the cell. Whenever you have less solute, we call that, and there's more solute on the outside of the cell. So whenever you have less solute, we call that area hypotonic. And whatever area has more solute, we call that hypertonic. So inside the cell, the condition is hypotonic because there's less solute in there, less red dots. And outside the cell, it's hypertonic because there's more solute out there, more red dots. Okay, less solute's always hypotonic, more solute's always hypertonic. And here's the key: water moves to the side that's hypertonic. So in this case, water is going to leave the cell. And if water leaves the cell, you can kind of think of it like air leaving a balloon. If the thing that's filling up the balloon, in this case water, if it leaves the cell, the balloon's going to get smaller, or the cell's going to get smaller. So we would expect this cell to shrivel up if you place it in a hypertonic solution, in a solution that has more solute than the amount that's inside the cell. Okay, let's do another one here. Just like last time, we're going to count how many water molecules and salt molecules are inside and outside the cell. So inside, there's 4 water molecules, 4 blue dots. There's 8 red dots, so those are the salts. Outside, there's 16 water molecules and 2 red dots, 2 salts. So now we ask ourselves, okay, which side has more solute and which side has less solute? And remember, in this case, the salt is our solute. Water is always the solvent. So I have more solute inside the cell, less solute outside the cell. Wherever there's more solute, that is hypertonic. Wherever there's less solute, that side is hypotonic. So water always moves toward the area that's hypertonic, where you have more solute. So in this case, water is going to move from outside the cell to the inside of the cell, because water moves toward the side that's hypertonic. But if water moves inside the cell, then imagine that kind of like blowing up a balloon. The cell's going to get bigger and bigger and bigger until you reach equilibrium. Okay, last one here. Inside the cell, I have 10 water molecules and 5 salts. Outside, I've got 10 water molecules and 5 salts. So notice we can ask ourselves which side has less solute, which side has more solute. Well, in this case, it's equal. When you have equal solutes on both sides, we call that isotonic. So this cell is isotonic with the solution that it's in. When you have a case that's isotonic, then cells will remain the same size because water is going to move in and out at an equal rate. So it's not like water's only going to come in or water's only going to go out. Water's coming in and out, and it's doing so at an equal rate. So the cell just stays the same size. Okay, so you've seen how cells maintain homeostasis through diffusion and osmosis.